So we classify risk by the set of possible outcomes as well as how likely each of those possible outcomes are to occur. So when it comes to GMOs, how do we measure the likelihood of these possible outcomes and the possible things that can happen? Tell us about this in some way that non-scientists can understand.
Sure. So scientists test and they test and they test, and sometimes for decades, it turns out. So GMOs have been in the lab for 30 years, and they've been on the market for at least 20 years. And since then, more than 1,700 peer-reviewed safety studies have been published, including a number of very detailed and lengthy reports. What's more, most reputable science organizations around the world agree that existing GMOs as we know them, are no more risky than their conventional crop counterparts. In fact, these are the most tested foods in the history of mankind.
It's impressive.
That's right. So GMOs are extensively tested long before they go to market for both their potential impacts on the environment as well as for their food safety. So the desire to protect the health of the environment and the health of people is really the primary driver for all of this government oversight.
So let's go back to the case of the GMO papaya. Tell me how this worked in that case.
Sure. So many tests were done to ensure that the technology would have no adverse effects on the environment. So, for example, the researchers tested to see if there might be differences in the soil and among the microbial populations around GMO and non-GMO trees. Researchers also looked into whether there might be adverse effects of GMO papaya on beneficial insects, so insects that are pollinators or predators of unwanted insect pests. They also looked at the likelihood of gene flow, so the likelihood that pollen from an outcrossing GMO plant might actually reach and fertilize a non-GMO plant. Other tests look at the impact that a GMO crop may have on any nearby relatives. And weediness is also looked at. So we want to ensure that the GMO crop has no impact on other plants in the environment, which may actually confer some kind of advantage.
So that tells us some bit about how we protect the environment. What about food safety?
Sure. So GMOs are usually first tested to assess whether they're substantially equivalent. That is to say that their food qualities compared with non-GMO counterparts are the same. There are no differences in their nutritional profiles, and there are no toxic molecules present. Studies are also done to look at any potential allergens that might emerge from reshuffling the genetic code. So if allergen signatures or compositional differences are detected, of course, further studies have to be done to determine their safety.
What is required to ensure that it is safe enough that it could be taken to market?
Well, that depends to some extent on each country's regulatory system that they have in place.
So, for example, in the U.S., what would be required?
Right. So in the U.S., the regulatory system is overseen by 3 independent agencies: the United States Department of Agriculture, the USDA, the Environmental Protection Agency, the EPA, and the Food and Drug Administration, the FDA. And each of these agencies oversees different aspects of GMOs. So the FDA, for example, ensures the safety of human food and animal feed, as well as proper labeling, and the safety of all plant-derived foods and feeds. And although the FDA reviews the safety of genetically engineered animals meant for food and also for feed, consultation with the FDA on safety of GMO crops is actually in the U.S. voluntary, but it is routinely sought. The EPA, on the other hand, regulates pesticides, and this includes plant-incorporated protectants. So this would include Bt genes, for example, or even that coat protein that was used in the case of the GMO papaya. APHIS, on the other hand, regulates the introduction of certain GMOs that may actually pose some kind of risk to plant health.
So an APHIS, that's the Animal and Plant Health Inspection Service, right, of USDA.
That's right. So going back to the case of the GMO papaya, approval was sought from all 3 of these agencies. So APHIS was concerned with crossing with other wild relatives. The EPA was concerned that the coat protein was derived from a plant pest. And the FDA wanted to ensure that the papaya was nutritionally equivalent to non-GMO papaya. So scientists had to address all of these concerns.
So if it's set up so there has to be sort of this acceptable level of risk, that suggests that there is still some risk, that some bad things can happen. What sorts of things can get through?
So the U.S. regulatory system is pretty robust, and of course researchers do have it in their best interest to ensure the safety of their own work. So for example, in the mid-1990s, there were some researchers who were trying to make soybeans more nutritious by enhancing for an amino acid that is normally lacking in soybeans. So they introduced a gene from the Brazil nut. And after doing this, they found that this particular gene turned out to be a likely allergen in the Brazil nut. And with that finding, they immediately stopped doing this research, and it never even made it into review by regulatory agencies. The research was stopped. So one of the criticisms of the anti-GMO advocacy networks is that we never really know how long is long enough.
Yeah.
So for how long should we be conducting these tests before we can say with certainty that something is safe? One of the limits of science is that we can only comment on the evidence at hand. So this is why politics and activism often trumps science. While science can only say that to date there is no evidence that something is unsafe, activists and conflict entrepreneurs are not bound by this limit of science. What is important is to look at trade-offs. So for example, if we know that a GMO can displace pesticide use, and we know that that pesticide is dangerous, any unknown risk of the nature of the GMO itself, one can argue, may be worth taking.
From the point of view of an economist, we would also be worried with the possibility of false positives, that somehow through this process we end up detecting a hazard or a risk that's really not there, and it prevents good products that provide us some sort of benefit from actually coming to market. And to tell you a little bit about how something like that could happen, usually in these processes there are actually several different phases that we're concerned with. So for example, with a pesticide, you're concerned with the probability a worker is exposed to it sometime in the field. But then you're also worried about the next phase. So given that they're exposed, what's the probability that they face some sort of adverse effect from that exposure, or something toxic from there? And the way this standard tends to be applied is that we look at each phase separately, and we apply this probability standard first to the exposure, and then also we apply it to the possibility of the adverse effect. And at each phase, we're sort of rounding the probability of the bad outcomes up, so that we're making sure we're as safe as we can be. But this leads to something called creeping safety. So first we're rounding the probabilities up, but then when we start looking at the overall risk, you have to multiply these probabilities together and they compound. So what happens is, if you happen to violate that threshold of safety at any phase, even just because you round it up, you're left out, right? And it's actually possible that things that meet the overall standard of safety wouldn't actually pass the test because of those individual phases.
So this could result in perfectly safe products with explicit consumer benefits never actually even making it to market.
Exactly that. Exactly that. Now, bringing this back to our original question, to make good decisions about risk, we sort of need the list of possible outcomes and the probability associated with each one of those outcomes. Is that what we get out of this process?
Yes and no. So the process is focused on finding how likely we are to see some set of known possible bad outcomes. It's not exactly a list of all possible outcomes, because we actually ignore some negative outcomes that we might not normally test for. Moreover, we don't exactly obtain probabilities. We more just figure out if there's a substantive chance, maybe a ballpark guess, at the probability of various extents of bad outcome.
And that's not to mention all the possible neutral or even beneficial outcomes that are sort of ignored in this process.
That's right. The original research tries to anticipate risks, but really assesses the benefits. It's the regulatory process mainly that's concerned with risks and potential novelty.
If we had to summarize, I guess what we'd say is, while we don't have a perfect system in place, we do have one that produces regular results and ensures our safety. So if you had to choose, what would you consider too unsafe to make it to market, and what would you consider beneficial and safe enough that we really should bring it to market?